Hi everyone, welcome back. In this section, we're covering Chapter 16, Section 16.3, Modes of Disease Transmission. This is one of the most clinically relevant parts of epidemiology because preventing transmission is the foundation of infection control. By the end of this lecture, you should be able to describe the different types of reservoirs, compare contact, vehicle, and vector transmission, identify major disease vectors, and finally, explain why healthcare-associated infections occur so often. So let's begin by looking at where pathogens come from. To understand how diseases spread, we need to know two things. One is where the pathogen lives. That's called its reservoir. The second is its mode of transmission, which is how it moves between hosts. Some pathogens spread really easily, while others require very specific conditions. We're going to talk about all of these. And vectors, for instance, these are animals, usually some kind of insect or tick, that carries and transmits a pathogen to humans. And remember, generation time, we learned this a while ago, that's the average time it takes for a population to reproduce and for a new generation to emerge. So because pathogens reproduce much faster than we do, they adapt quickly to new environments. A reservoir is the natural habit of a pathogen, and it can be living, such as in humans or animals, or non-living, such as soil or water. For some examples of non-living reservoirs, we have Clostridium tetani, remember that bacterium that causes tetanus, can survive in the soil as endospores. And in water, we might find bacteria like Salmonella, or the bacteria that cause cholera, Vibrio cholerae, or even viruses, especially those that infect our GI tract, like enteric viruses. And some pathogens, scarily, can persist on surfaces for days, like influenza virus. Our book gives us an example of a study that found that influenza virus could survive on money, like think of a dollar bill, for up to 17 days. So reservoirs maintain the pathogen between infections. So humans can be reservoirs, and in some cases, humans can even carry and transmit pathogens without symptoms. So carriers are people who harbor and transmit pathogens, and we have three types here. A passive carrier is not infected. They're simply intermediates that transfer microbes from contaminated hands or surfaces to someone else. An example from our book is a healthcare provider who fails to wash their hands after seeing a sick patient. So they pick up the pathogen from that patient on their hands, and they don't wash their hands, and they transfer those pathogens to the next patient, making them sick. An active carrier is infected and contagious, and an asymptomatic carrier is infected but has no symptoms, yet they can still spread the disease. A very classic example of an asymptomatic carrier is Typhoid Mary or Mary Mallon. She was the first person in the US identified as an asymptomatic carrier of the pathogen associated with typhoid fever. So for typhoid fever, more commonly known as just typhoid, it's a bacterial disease that you get by ingesting food or water that's contaminated with the feces of an infected person that contains the Salmonella bacterium. Unfortunately, Mary Mallon was a cook. That was her career, and she was asymptomatic, so she actually is connected to a lot of cases, around 51 cases of typhoid fever, including 3 deaths. So Mary Mallon's background is pretty interesting. She worked as a cook for wealthy families in the early 1900s. And what happened was the families she worked for started getting sick. And so as soon as that happened, she started to leave those families. She would leave in the middle of the night, find a new family to work for. Eventually, one of the families in 1906 hired George Soper, who was an expert back then in typhoid fever epidemics, to try to figure out what was causing them to get sick, causing those family members to get sick. So eventually he tracked Mary Mallon down and directly linked many cases of typhoid fever directly to her. The New York Health Department got involved, and they found typhoid bacteria in her gallbladder. They also interviewed her, and she admitted she had poor hygiene. So she didn't wash her hands when she was cooking, and she did not understand how she could be the one transmitting typhoid because she was perfectly healthy herself. And, back then you have to remember that they didn't understand microbes very well. She felt perfectly fine, and I can see why it would be confusing for her for people to accuse her of transmitting typhoid. So she refused to get her gallbladder removed, and so they isolated her for 3 years, eventually releasing her as long as she promised not to cook anymore. So she did become a laundress for a while, but that didn't make enough money compared to being a cook. So what she did was she changed her name and started working as a cook again. But again, what happened was as soon as she started working as a cook, more cases of typhoid came up, more people died. They found her again, and they put her in confinement for the rest of her life. And if you're interested, I cut and paste this directly from our book. That gives us a link to George Soper's direct account on his work with Typhoid Mary, with Mary Mallon. Many years ago, something similar happened in the U.S. with nurse Casey Hickox, who was a nurse working with Doctors Without Borders, who had treated Ebola patients in Sierra Leone in 2014. So when she came back to the U.S., she had no symptoms when she landed at Newark Airport, but she was detained in New Jersey and forced into a quarantine tent, a mandatory quarantine for about 80 hours against her will. She was not allowed to leave. When she was finally released, I think to her home, I believe it was in Maine. Yep, it says Maine. She was ordered to stay home for 21 days and not go to public places like subways, buses, or parks, but she refused, citing evidence that asymptomatic people are not contagious. So this case was important because it highlighted the conflict between state orders versus CDC guidelines and public fear versus science-based risk assessment. So I think the core message for the Mary Mallon case is that asymptomatic carriers can be major sources of disease spread, and lack of cooperation can force strict public health interventions. On the other hand, public health actions have to balance safety with individual rights, right? And you have to follow scientific evidence and not just fear. Animals can also serve as reservoirs, and diseases that spread from animals to humans are called zoonoses. For zoonoses, transmission can be direct through a bite or a scratch, or indirect through vectors or the environment. So remember, for parasites, we have a definitive host. This is where sexual reproduction occurs, and an intermediate host that carries immature or asexual stages of the parasite. For example, in Plasmodium, remember this is a protozoan that causes malaria, the female Anopheles mosquito is both the vector and the definitive host because Plasmodium reproduces sexually inside the mosquito. Our next checkpoint asks, list some non-living reservoirs for pathogens. So non-living reservoirs are environmental sources that support pathogen survival outside of a living host. Examples could be soil. Remember soil, we can find Clostridium tetani that causes tetanus, where they can survive as resistant endospores. Water can hold pathogens like Vibrio cholerae that causes cholera, or Salmonella typhi that causes typhoid fever, or surfaces and objects. So like our book mentioned, influenza virus can survive on money, dollar bills, for days, depending on the conditions. For the second checkpoint, they ask, explain the difference between a passive carrier and an active carrier. So a passive carrier is not infected. They simply carry the pathogen on their body or surfaces. An example was a healthcare worker who didn't wash their hands between patients and transmits bacterium between the patients. An active carrier, on the other hand, is infected, and they're capable of transmitting the disease to others, and they may or may not show symptoms. For example, someone can spread influenza, spread the flu, before or after they have symptoms and signs. And then we mentioned also asymptomatic carriers. That's a type of active carrier that shows no symptoms but can still spread the disease, such as we saw with Mary Mallon and Salmonella typhi. So these distinctions will help you recognize the different sources of outbreaks. Regardless of what the reservoir is, transmission has to occur for the infection to spread, and transmission from the reservoir to the individual, animal or human, has to occur. Then the individual has to transmit that agent, that pathogen, to other susceptible individuals, either directly or indirectly. So there are a variety of different types of transmission mechanisms, unfortunately for us, right? There are three major categories of transmission routes, including contact transmission. This is going to be between individuals, either directly or indirectly, and we're going to go over each of these in detail shortly. It can also be through a vehicle, like water, air, or food, or through vectors, which are usually some kind of bugs that can transmit the pathogen as well. Each of these has a clinical relevance in preventing the spread of infections. So let's look at contact transmission first. And contact transmission can be direct or indirect. For direct contact, we're looking at physical interaction directly between two hosts. In the subtypes of direct contact, we have vertical, horizontal, or droplet. In vertical contact, it's the mother passing the pathogen to the fetus or newborn, and this can be through the placenta, if it crosses the placenta, through childbirth, or through breastfeeding. So this is from one generation to the next for vertical direct contact. For horizontal direct contact, it's from person to person through direct touching, kissing, or sexual contact. And droplet is also considered direct contact as long as it's less than 1 meter in distance. And this can be through coughing or sneezing, like we kind of see here, which easily transmits diseases like the flu or pneumonia. Indirect contact is through fomites. Remember, these are non-living objects that have been contaminated with the pathogen. For example, a doorknob, maybe someone's coughing into their hand and then they touch the doorknob, through dirty towels, needles, catheters, or surgical tools. And this figure emphasizes that pathogens often enter through mucous membranes. So you might touch a contaminated surface with your hand, but then you touch your eyes or your nose or your mouth, creating this route of entry. This is why hand hygiene is a really critical part of infection control. Washing your hands can prevent many types of infections. Fomites are involved in indirect contact transmission. So fomites, remember, are non-living objects, and they can spread pathogens easily in hospitals, schools, or public spaces. Dirty doorknobs, dirty shared towels or books, even stuffed animals, needles and catheters especially are significant because they bypass the skin barrier in hospitals and clinics. Here's an example. Unfortunately, that's happened more than once in the news. One of the more recent cases happened in spring of 2025, where a nurse was reusing the needleless part of the syringe. So we have seen syringes before, and you can see the needle is attached at the end, but there has been more than one case where nurses or other healthcare providers, including doctors, have reused this part of the syringe, and this can act as a fomite for indirect contact transmission. All right, so far we've covered contact transmission, including direct and indirect contact transmission. Our next type of transmission is vehicle transmission, and this involves a non-living medium and can be water, food, or air. For waterborne transmission, this involves microbes in contaminated water, such as the microbes that cause cholera or typhoid. For foodborne transmission, this is usually from improperly handled or improperly stored food and can carry microbes such as Salmonella or E. coli. And for airborne transmission, this includes fine aerosols that can travel more than 1 meter. Remember, less than 1 meter was direct transmission. More than 1 meter, now we consider this airborne transmission, which is a type of vehicle transmission. Tuberculosis, or Mycobacterium tuberculosis, the microbe that causes tuberculosis, is a classic example and requires special ventilation and masks. Tuberculosis is often transmitted through airborne transmission, so airborne, when the causative agent, the bacterium, Mycobacterium tuberculosis, is released in the small particles that come out with our coughs. And because it's pretty scary, tuberculosis requires as few as 10 cells to initiate a new infection. Patients with tuberculosis have to be treated in rooms that are well equipped with special ventilation, and anyone entering that room should wear a mask. Here we see an example of food, which could also be a vehicle of transmission, and they have a glass shield here to prevent respiratory droplets from falling onto food. Many gastrointestinal pathogens spread through food if food is improperly protected. And going back to our clinical example from the first two videos for Chapter 16, remember that Salmonella outbreak from those contaminated turduckens? This is a classic foodborne vehicle transmission event. So the vehicle transmission was through that food, the contaminated turducken. So public health officials identified the source and removed products from circulation in order to stop further spread. Our third type of transmission is vector transmission. So far we've had contact transmission, and then vehicle transmission, and then again now we have vector transmission. So vectors are living organisms, usually arthropods, such as mosquitoes, ticks, lice, mites, or fleas that can transmit pathogens, but can also include non-arthropod vectors such as mammals who can transmit disease such as rabies, or birds that can transmit avian flu. Of the vectors, there are two subcategories, including mechanical and biological vectors. Mechanical vectors carry the pathogen on the surface of their body. They're not actually infected themselves. A nice example from our book is flies flying around. Maybe they land on feces and pick up some fecal bacteria, and then they fly around further and maybe land on food on a picnic. All right, someone's having a picnic and the flies land on that food. That can transfer the bacteria from the feces to the food and make someone sick. Biological vectors actually have the pathogen inside of them, and the pathogen is reproducing or developing inside that biological vector. So one example is in mosquitoes. So mosquitoes are an example of a biological vector. They can carry plasmodium, and then they can bite the next person and transfer the plasmodium to the human to cause malaria. Ticks are also another type of biological vector, where Borrelia burgdorferi, the bacterium, can be within the tick, so they're developing within the tick, and when the tick bites the human, that bacterium can be transferred to the human to cause Lyme disease. In this figure, we see an example of both a mechanical vector on the left as well as a biological vector on the right. So a fly, like a housefly, is a mechanical vector. It can pick up bacteria on its feet and then transfer them to the food. Whereas in biological vectors or biological transmission, such as malaria or Lyme disease, the vector itself becomes infected and transmits the pathogen through biting. Figure 16.13 in our book shows common vectors, and it's a little small here. It's hard to see, so I'm going to zoom in the next slide. Here I'm zooming in the top half of that table or figure, and I can see, for example, the flea. This is the genus and species name for the flea, Synopsilla chiopis. So this is a vector for, let's pick one, let's pick plague. So plague is caused by the bacterium Yersinia pestis, and fleas can transmit plague by carrying Yersinia, this bacterium, as an intermediate host for the bacterium. And here's that bottom half of that same figure. Mosquitoes can be vectors for several different types of diseases, including malaria, dengue, Zika, West Nile virus. Ticks can transmit Lyme disease and Rocky Mountain spotted fever by carrying these bacteria, by being vectors for these bacteria. And the tsetse fly we saw in lab can transmit sleeping sickness by carrying trypanosoma. This is a type of protozoan. So vector identification is essential in outbreak investigations. And what do you think all of these vectors were? Were these biological vectors or mechanical vectors? So all of these were biological vectors. This checkpoint asks, describe how diseases can be transmitted through air. So this is airborne transmission, and that occurs when small particles or aerosols containing pathogens are carried over long distances. Remember, this is more than 1 meter. For example, Mycobacterium tuberculosis can be spread if you inhale droplets from others who have coughed or sneezed. Another example I didn't talk about is hantavirus. This can be actually spread by inhaling dust that has been contaminated with dried rodent urine or feces. And factors that affect air transmission include air currents, humidity, and the size of the particles. The smaller the droplet size, the longer they stay suspended in air. So we can prevent airborne transmission with proper ventilation, air filters, and wearing masks, especially in healthcare settings. For the second one, explain the difference between a mechanical vector and a biological vector. I'm going to show you a table in the next slide. So here's that table that we saw a few slides ago, looking at mechanical versus biological vectors. And here I've blanked out the description and examples to see if you can fill them in. Remember that mechanical vectors do not become infected. Biological vectors do. So only biological vectors support part of that pathogen's life cycle. So the key difference, or one of the key differences, is in that biological transmission, the pathogen is going to live and often, but not always, multiply within the vector before it infects the next host. And in this section of our book, they highlight an ethical issue using genetically modified organisms, or GMOs, to control vectors. So one example is by what they've done is release genetically modified sterile males of Aedes aegypti and Aedes albopictus mosquitoes to reduce the mosquito population. And there have been many pros. There was a significant reduction in the number of dengue transmission cases, and they were able to specifically control the species of mosquitoes. But cons included risks or concerns, such as concerns that the gene could escape the species boundary of Aedes aegypti and cause problems in other mosquito species. Maybe that could lead to unforeseen ecological consequences. Also, this particular study was administered by a for-profit corporation, so there might be additional conflicts of interest. It was also not clear how any unintended consequences of this program could be reversed. So this is a good example that demonstrates the delicate balance between public health and environmental ethics. This figure shows the range of Aedes mosquitoes that transmit Zika. So climate and habitat expansion really influence the distribution of vectors, which in turn affects disease risk. So Zika virus infection during pregnancy, for instance, can lead to very serious birth defects and developmental problems. The most common defects are microcephaly, so you'll have an abnormally small head and brain, and other very severe brain abnormalities. But it can also cause eye defects, hearing loss, joint and limb problems, and seizures. And these issues can be present at birth or develop slowly as the child grows. Interestingly, Zika is also one of the smallest viruses out there. Here I can see it on this relative scale at 0.045 micrometers. So it's even smaller than bacteriophage, which are viruses that infect bacteria. Quarantine restricts the movement of exposed or infected individuals. And we can also isolate patients. Isolation separates infected individuals, and both have been used for high-risk pathogens such as tuberculosis, Ebola, and during the COVID-19 pandemic. Quarantine decisions really have to consider incubation period and likelihood of transmission for the diseases. This slide shows modern quarantine measures, including specialized transport units and isolation wards used for Ebola. And the purpose of these systems is to protect healthcare workers and prevent community spread of the disease. Healthcare-associated infections, or HAIs, are sometimes also called nosocomial infections. These occur in hospitals and healthcare settings due to invasive procedures or immunocompromised patients. There are also a high density of pathogens and possibly contaminated equipment in these hospitals and healthcare settings. So common HAIs include surgical site infections, pneumonia, urinary tract infections, and bloodstream infections. So one of the most important methods to prevent this is hygiene of the hands, so washing hands, and sterilization of the equipment. So these are critical preventative measures. And in the book, they provide us with a little older CDC values of HAIs. So this was the rate back in 2011. Today, they use a different type of reporting data. They usually report by category these days, and they're called standardized infection ratios, where they look at select healthcare associated infection types, like central line associated bloodstream infections or catheter associated urinary tract infections. So it's hard to compare recent data with older data. All right, some key takeaways from this section. We saw that reservoirs sustain pathogens between infections. There were 3 types of transmission routes: contact, vehicle, or vector. We saw quarantine and infection control can prevent the spread of disease, and understanding how a disease is transmitted can really help us create interventions and prevention protocols. This checkpoint asks us to give some reasons why HAIs occur. So healthcare-associated infections can occur due to invasive procedures. Surgery, catheters, or ventilators can allow microbes direct entry into the body. Compromised immunity is also important. Hospitalized or elderly patients are generally more susceptible to infection. There could also be cross-contamination, so inadequate hand washing or improper sterilization of instruments can cause cross-contamination. Environmental factors include the presence of high concentration of pathogens in hospital settings, including opportunistic pathogens. So normally harmless microbes can exploit a weakened immune defense system. So prevention is important. Strict adherence to infection control protocols, aseptic technique, and regular staff training is important. All right, let's recap 16.3. The first one described different types of disease reservoirs. Remember, reservoirs are natural habitats where pathogens live, grow, and multiply when they're between infections. For the types of reservoirs, we had human reservoirs. These can include infected individuals or carriers that are symptomatic or asymptomatic, such as typhoid Mary. We have animal reservoirs that transmit zoonoses, so animals can transmit pathogens to humans through direct or indirect contact. Bats can transmit rabies, for instance, or birds can transmit bird flu. Rodents can transmit the plague. Non-living reservoirs were things like soil, water, or contaminated surfaces that harbor pathogens. For instance, soil could carry the Clostridium bacterium through its endospores. For the second one, I'm going to show you in the next slide through a table. So let's go to the third one, important disease vectors. Vectors are living organisms that transmit pathogens between hosts. Arthropods are the most common type of vector, including mosquitoes. Mosquitoes are probably the most prevalent vector because they transmit diseases like malaria, dengue, Zika, or yellow fever. Other examples of vectors included ticks, lice, fleas, or mites. There are also non-arthropod vectors like mammals that can transmit rabies, or again, the birds that can transmit bird flu. For the prevalence of nosocomial infections, so nosocomial infections, also called hospital-associated infections, are infections acquired in hospitals or healthcare, sorry, I said hospital, but healthcare-associated infections. They can be acquired in healthcare facilities. So prevalence was from 2011 in the book. They reported over 720,000 cases. And the reasons for these HAIs were high density of immunocompromised patients, frequent invasive procedures, cross-contamination, and prevention. Important ways are by washing hands, sterilizing equipment, practicing aseptic technique, and frequent training of the staff. All right, going back to that second learning objective, looking at contact, Vehicle and vector transmission. Contact transmission was between hosts, such as direct or indirect contact through fomites. Vehicle was through water, food, or air. And vector transmission was usually through those arthropods, which can occur through mechanical or biological transmission. That concludes our third video for Chapter 16. In the next video and final video for Chapter 16, we're going to look at global public health systems and how we respond to emerging diseases.